Hello, wonderful AP Bio students, and welcome to our video on mutations. You've probably heard me say this before, but you are a mutant. And so am I. I'm actually more of a mutant than you are because I'm older than you, just a tiny bit older than you. I potentially have more mutations than you. Now, in society, we've seen mutations as kind of a cool thing. We have the X-Men, be able to freeze water, to be able to jut out metal swords from your knuckles. It's pretty cool. Those aren't real, but it's an interesting concept for sure. And we have some mutations that are bad, like cancer. And then we also have mutations that give us interesting phenomenon, like this boy right here. This boy is this man's son, and this is a picture taken in Papua New Guinea. And this boy has a genetic disorder called albinism, which makes him not produce melanin correctly. And melanin is a protein in your skin that gives your skin its darkness. So people that have darker skin have more melanin. And the purpose of melanin is to protect your skin from UV light. So this young boy is entirely genetically this man's son, but he doesn't have the same physical characteristics as him because of his mutation causing albinism. So today in our video, we're going to talk about what a mutation is, what causes it, and some types of mutations that can lead to some common genetic disorders. But one thing that's really important to remember is that mutations can be both good and bad. So we hear a lot about the bad mutations like Tay-Sachs disease and cystic fibrosis and even lactose intolerance. Fibrosis is a bad mutation because you can't enjoy ice cream like all of us non-lactose intolerant folks. And the thing about bad mutations is they become really bad when they are a mutation in a sperm or an egg cell. And if we have a mutation in the sperm or an egg cell, then it can be passed down to the next generation. But then we want to look at the other side, some examples of good mutations. There are very few good mutations, but they do happen. And this is an example of one. This is a kind of an artist's rendition of the HIV virus. And there's a mutation in this gene that you have in all of your T cells, which T cells are part of your immune system, called the CCR5 gene. And if that gene is mutated, then the HIV virus cannot infect those cells. It's thought that about 1% of the population actually has this mutation. So yes, mutations can often be bad, but sometimes they're good and allow for somebody to survive something that somebody else wouldn't. So let's get a general definition of mutation. A mutation is simply just a change in DNA. So it's a permanent change in genetic material. Now, sometimes you can have changes in your genes, but your DNA has those built-in fix-it mechanisms. Well, mutation is a permanent change in genetic material, and they are spontaneous and they are random. And so if we look at these strands of DNA, so here we have, these aren't complementary, obviously, but here we have some DNA strands. A mutation can be as simple as just the change of one nitrogenous base, or it can be the change of an entire base pair. So let's look at what causes mutations. Mutations are caused by mutagenic agents, and they can occur in either somatic, so the word somatic means your body cells, so pretty much all the cells in your body except your gametic cells, and your gametic cells are sperm and egg. So mutations can happen in either of these, and they can happen one of two ways. One, DNA is copied incorrectly. Well, you just learned about DNA replication, and you know that there's all of these enzymes that check and make sure that the DNA is copied correctly. But let me put this in perspective. In your lifetime, you will have 10,000 trillion cell divisions. That is like so many cell divisions. That is so many to have 100% accuracy in that DNA replication. Also, some recent research has come out that DNA has damages that are naturally occurring. So just from being a human in this world, we have UV light, we have heat damage, we have burned skin. Just by being functioning in daily life, DNA is naturally damaged. And this number really blew me away. So naturally occurring DNA damages are more than 60,000 times per day per cell. So that means that those little mechanisms that we learned about that check DNA and fix DNA, fix 60,000 damages per day per cell, which just blows my mind. Another way that DNA can be damaged and cause a mutation is if it is harmed from exposure to radiation or some sort of chemicals. And we know that anything that causes cancer, and we know that radiation and chemicals, a lot of them can be cancer causing. We call them a carcinogen. And those are just cancer-causing agents. And in the long run, cancer-causing agents are mutagenic agents. So let's take a look at what really mutation means. So mutation causes proteins to be produced differently. They can be produced as a different shape. They can be not produced at all. So in some way, it makes them function differently or not function at all. And the crazy thing, well, a lot of times we think about it in a bad way. Like, we want all of our proteins to work, obviously. But if we have a mutation in a gene and it gets passed down, then it essentially introduces new genes into the population. Some of those genes are bad and some of those genes are good, but mutation gives us this word variation. It gives us our species and all species of the world more genes to access. So mutations give us kind of that raw material for evolution, that raw material for natural selection to add on. I want to talk about a built-in mechanism that you have and all life has to avoid a lot of damages from mutation. And this built-in mechanism is called the wobble. I don't know why they didn't think of a more scientific name for this, but I kind of like the fact that it's the wobble. So if we look at, this is an amino acid wheel. We have our 20 different amino acids, and this wheel helps us decipher codons. So if we have the codon AUG, we can look at this wheel. We see A, we see U, and we see G, and we say, oh, AUG is the codon for methionine. Methionine is actually always the start amino acid, but you guys knew that. So the thing about wobble is that if we look at an amino acid, for example, like valine, the first codon has to be G, the second codon has to be U, but the third codon can be G, A, C, or U. So it could be GUG, GUA, GUC, GUU, and that all codes for valine. And so if we have a mutation in this third nucleotide in the codon, then it's not going to affect the order of amino acids. It's still going to be valine. Now, some amino acids are more wobbly than others. For example, serine is another that can have 4 different codons coding for serine. But if you look at tryptophan, the only thing that codes for tryptophan is UGG. So tryptophan is actually a less versatile amino acid in that if there's a mutation in the codon for tryptophan, there's no chance that it can still be tryptophan. It's going to be something else, like cytosine or stop or tyrosine. So that is wobble. Now let's take a look at a few different types of mutations. The simplest type of mutation is a point mutation. And this is simply when a single nitrogenous base pair is substituted for another. For example, if this is our original DNA, and on this spot we had cytosine and guanine, and swap, now all of a sudden we have thymine and adenine. So if we just take one nucleotide pair out and replace it with another, and if that mutation becomes permanent, if it stays undetected, then bam, you are a mutant. Now, the crazy thing about that is you have so many of these nucleotides that a point mutation, one single point mutation, can lead to a lot of disorders. I'm going to talk about a few. But you have 3 billion base pairs. So if you have a point mutation in one single one of those base pairs, it could potentially lead to a disorder. Some examples of those disorders are progeria. Progeria is when one cytosine is replaced with a thymine on the 1824th base pair of a gene for lamin. So basically lamin A is a gene with a ton of base pairs, and on the 1824th base pair it should be cytosine, but instead it's thymine. And if it's thymine, then your lamin A protein, which provides structural support for the nucleus, does not work anymore. And if we look at what that means for the cells, we have a normal healthy cell here. Progeria cells seem to be a little deflated and have less structural support. And progeria is known as the rapid aging disease. And we learned a bit about progeria. This is Sam. He passed away just recently. He was 17 and had progeria. But progeria causes hardening of blood vessels. You have issues with cardiac. You have issues with bone density. So it's a disease where the life expectancy of somebody with progeria is about age 13. So another example of a point mutation is Tay-Sachs. And Tay-Sachs is a point mutation on your 15th chromosome. And that causes this enzyme that is coded by the hexagene, causes this enzyme to be misshapen and not work. And this enzyme breaks down a special type of lipid. And because those lipids cannot be broken down in your body, these lipids accumulate and actually disrupt how neurons pass signals to each other. So it causes basically premature death of nerve cells. And it's really, really rare to have a child with Tay-Sachs disease to live past 6 months. So this is a very serious, very life-threatening disease to have. One other example is cystic fibrosis. You have these channel proteins that are specifically coded for to pass chlorine ions in and out of cells, specifically in the lungs. And so number 1, this is normal. This is your normal channel protein. See, it's working fine. Chlorine ions can move in and out. In cystic fibrosis, that channel protein is not functioning and cannot move those chlorine ions. And the symptoms are you get cysts in the pancreas, difficulty breathing, which is the most severe symptom, lung infections, and this really interesting thing called clubbing, or watch glass nails, where your fingertips get really enlarged. So that is cystic fibrosis. Let's look at another type of mutation, and that is insertion or deletion. So when we're talking about insertion or deletion, we're talking about one single nucleotide. And we know that a codon is how many letters? Say it right now. How many letters is a codon? Codon is 3 letters. So basically, our body only understands 3-letter words. You give it a 4-letter word, no comprende. Give it a 2-letter word, no comprende. So it's really important that we keep our 3-letter words good and accurate. So I have a few 3-letter words here that kind of make a coherent sentence. The big cat was red. So an insertion is when is exactly what it sounds like, is when a nucleotide is added somewhere. So let's say we add, I'm just going to pick a random one and see what we get. We added the letter N here. So since we can only read 3-letter words, we get the big can, toi, sre, de. Which the sentence starts out fine, right? But as soon as that insertion occurred, then everything after it, so the insertion occurred right here, everything after it is incorrect. So that's an insertion and deletion. If we get an insertion of 3 nucleotides, then that's a different story because our bodies can read that 3-letter word. Let's look at a deletion. It's pretty much the same as what it sounds like. If we take a nucleotide and we remove it, let's remove this G. Well, now instead of our sentence reading the big cat was red, our sentence reads the big at azer ed, which isn't at all what our sentence was. So once again, because we can only read those 3-letter words, it messes up everything from the point of the mutation on. Let's do a real example instead of a sentence. If we look at this right here is our normal strand of DNA, and we see that the codons read TAC, GGA, CAA, TAC. And remember, each of these codons codes for one specific amino acid. So if we insert a C to that, and we read off our codons, we get TAC, GGA, all good there. CCA, well, it was supposed to be CAA. That's most likely going to code for a different amino acid, especially since it's earlier on in the codon. And if we look at a deletion, instead of reading TACGGA, our deletion goes TACGGC. It skips that A and goes straight to the C. So this all in all causes what is called a frameshift mutation, which is when nucleotides are added or deleted, the number not divisible by 3, because remember, we can only read those 3-letter words. And so because RNA is read in triplets, or codons, the reading frame is shifted, resulting in a completely different translation. So wherever that mutation was, so everything's good here, right? Wherever that mutation was, if this is our mutation, from this point on, this is my version of a messed up DNA. From this point on, the genetic code is going to be mutated. It's not going to be the same. So we really don't want frameshift mutations to occur. If we look here is yet another example. We have ATG coding for methionine, GAA coding for, I think this is glutamine, alanine, and glycerine or glycine. If we get rid of this G, then the only codon that actually stays the same is methionine. From that point on, everything is different. So then we get lysine and histine instead. Okay, an interesting example of a disorder caused by frameshift mutation is hypertrophic cardiomyopathy. And this is caused by a mutation in the gene coding for cardiac sarcomeres, which are important structural proteins in cardiac muscle. And an improper coding of that leads to this thickened left ventricle heart wall. And this thickened left ventricle heart wall is actually a cause of death. It's sudden death in young athletes. There's about 100 deaths per year in the U.S. caused by this hypertrophic cardiomyopathy. It's literally like a super young, healthy person who's an athlete gets cardiac arrest. And what's interesting is that in the U.S., you can't really get genetically screened for this as an athlete, whereas in Canada, you can, as long as you have some sort of hint of a family history of this disorder. So that brings up other issues of how we deal with genetic screening. Let's talk about another type of mutation. This is a nonsense mutation. It doesn't make no sense. It's nonsense. That is a point mutation in DNA sequence that causes stop to be coded for too early. And this results in an incomplete mRNA strand, and most likely, probably 100% likely, a non-functional protein. So let's take a look at this example. You can pause the video if you need to copy down this definition. An example of a nonsense mutation. This is our normal original DNA code. And we have the same amino acid coded for over and over again. Some proteins are like that. They're made up of a bunch of the same amino acid. And we take this C here, and we replace it with a T. And all of a sudden, we have one of the codons that code for stop. So this would be A, U, C, that code for stop. Well, this obviously looks like it keeps going, but this amino acid chain could be 100 amino acids long. But if we have a stop code early on, then our amino acid chain gets truncated. It gets cut off. And now we have this little tiny little short mini little amino acid chain that is pointless. It's worthless. It can't result in a functional protein. An example of a nonsense mutation is muscular dystrophy, and this is actually more common in boys because it's on the X chromosome, and it occurs in 1 in 3,600 boys. And what happens is there's a nonsense mutation in the code for this protein dystrophin, and this protein dystrophin provides structural support for muscle cells. And so you have severe muscle deterioration in these boys, and they lose a lot of function of especially their limbs and the ability to hold up their neck, and then later on in the ability to use their organs. So it's a really severe disease that luckily we found the gene for it, but it's a disorder that you definitely wouldn't want to pass on. Nonsense mutation is not to be confused with a missense mutation. A missense mutation is less severe, I would say, but could be equally as detrimental. So a missense mutation is a point mutation where a single nucleotide change results in a codon that codes for a different amino acid. So if our amino acids read that's an M, methionine, leucine, tyrosine, we could have a change of a codon, and instead it reads methionine, arginine, tyrosine. So changing just one amino acid, and we know that amino acids have different chemical interactions with each other. So if you change one amino acid, you can effectively change the shape of a protein and therefore its entire function. So let's look at another example of this. We have our original DNA code. We have a bunch of cats, cat, and cat codes for the amino acid histine. And so on our mutated strand, we have cat, and all of a sudden CCT, cat. Well, we still make our full strand. We still make our full strand of amino acids, but one of those amino acids is incorrect. And this may, or it may not, produce a malfunctioning protein. So, in review, we know that mutations are a part of life. I'm a mutant. You're a mutant. We're all a mutant. Some mutations are really good, like the ones that 1% of the population has that makes them resistant to HIV. There's also thought that many of the people that survived the Black Plague had a mutation that allowed them to do that. Also, mutations are the source of new genes, and this is a really important thing to understand as we tread on into our evolution unit, that we can't get new genes for natural selection to act upon without mutations. And DNA mutations can sometimes result in either the wrong order of amino acids, a truncated mRNA strand, so a really early stop, and sometimes they can result in nothing at all. And that's why you and I probably have a lot of mutations, but it's probably not an issue. So I hope you enjoyed that little talk about mutations, and I hope you have a wonderful day. Bye.